Okay, so the number of questions you can ask is determined by the number of times a person is presented. When you're first up there presenting, we just want to make sure you survive, right? So we're going to be very nice to you. We're not going to ask you tough questions. The second time you get up there, it's really your job to make sure that everybody in the audience understands what you're talking about. So you can ask anything about the presentation. The third time you're up there, people can challenge you. So you can actually try to find where Andrew doesn't know, like where something, he explained something wrong, or whether the researchers maybe should have done something else. Basically, any question that you can use to, or that you can ask to make Andrew look dumb is fair this time.
He's okay.
So.
I'm good on my own. All right. Just as a note before I get started, I'm going to try and work on making sure that everyone understands my presentation. So if at any point you have any questions about the presentation or what I presented, or if I'm talking too fast or too quietly, please raise your hand and let me know. I'm talking too fast. All right. With that aside, 33%, dear viewers, 33%. It's about 1/3 the odds of rolling a 1 or a 2 on a die, the odds of winning a car in the Monte Carlo problem if you don't switch, the odds of one of you falling asleep during my presentation. But more importantly, and on a more serious note, 33% is the odds of a breast cancer patient treated with tamoxifen suffering relapse at some point in the 5 to 10 year course of drug treatment. 33%. Imagine that you have just fought a battle with estrogen receptor positive breast cancer, one of the deadliest diseases known to mankind, and you are told by your doctor that the odds that you'll have to fight that same battle again, no guarantee of success, are 33%. And this is a conservative estimate, by the way. Some studies place the odds as high as 50%. I think that even a 33% chance of relapse is appallingly high and something that needs to be studied. Next slide. Sorry. So, but let me take a step back first. Hello, in case you weren't here when Mr. T introduced me, my name is Andrew Verdesca. I'm a third-time presenter and one of the managers and data reviewers here at Journal Club. And today I'll be presenting some work I did over the summer at NYU's Langone Medical Center under the guidance of my mentor, Philip. I'll be presenting on the probable causes of tamoxifen resistance in estrogen receptor positive breast cancer. That's a complicated title, but basically I'll be presenting on what I did this summer and why you should care about it. So because my work was mainly concerning breast cancer, I mean obviously we're going to have to talk about breast cancer. Breast cancer is the most commonly diagnosed cancer among women in the United States, and the second leading cause of cancer-related death worldwide. Obviously, breast cancer is a serious problem, and it affects lots and lots of people. Now, breast cancer is also one of the best studied types of cancer, and because of that, it's been classified by molecular markers, certain proteins, into 3 major subtypes: the ER/PR positive, HER2 positive, and triple negative subtypes. Triple negative breast cancer is defined by having neither of the 3 proteins, estrogen receptor, progesterone receptor, and human epithelial growth response factor 2. But for the purposes of my research and for the purposes of this presentation, we only looked at estrogen receptor positive or ER positive breast cancer. So we're only going to focus on that and forget about all the others. So what is ER positive breast cancer and what makes it different from all the other types? ER positive breast cancer, as the name implies, is characterized by the presence of the nuclear hormone receptor, estrogen receptor alpha. And approximately 70% of all breast cancers are estrogen receptor positive. This is really significant because estrogen receptor positive breast cancer is one of the most prevalent subtypes of one of the most prevalent cancers that we know of. And we really need an effective treatment for something this important and this dangerous. So what's interesting about estrogen receptor positive breast cancer is that the cancer is very dependent upon the estrogen receptor pathway, meaning that if you deprive the cancer of estrogen, or you cut the pathway that results in the expression of that results in cell growth, proliferation, survival through the estrogen receptor, you start to see the cancer stop growing and perhaps even start to shrink. Now this is really, really important and significant because a lot of drugs have been developed that target this particular pathway because it's so important to estrogen receptor positive breast cancer. Yep.
Like Lupron?
I don't know that particular drug, but sure. It could be, the problem is, there are a lot of different drugs out there that target estrogen receptor positive breast cancer. I'm not sure, well actually there are a lot of drugs out there that target breast cancer in general. I'm not sure if that targets estrogen receptor positive breast cancer specifically, if it's I don't know, sorry. Thank you though for your question. And anyone else, feel free to ask. Okay, so yeah?
What mechanism actually causes estrogen and the estrogen receptor to allow the cancer to survive?
I'm going to go over that right now. So basically the process by which estrogen causes estrogen receptor signaling is reasonably well understood. Androgens, which are a type of steroid hormone including testosterone, in the body get converted into estrogens with the aid of enzymes called aromatases. These enzymes in the individual cancer cells then associate with small proteins, these so-called cofactors, and bind to the estrogen receptor. Over there, the third one. Really need a laser pointer. And then this complex consisting of the estrogen receptor, estrogen, and those cofactors then causes the creation of transcription factors, which result in the creation of proteins such as survivin and c-MYC that result in cell growth, proliferation, and survival.
Could you do that last part of the question again?
Yes. Okay. So basically, let me re-summarize. Androgens get made into estrogens. Estrogens attach to the estrogen receptor and make proteins that are responsible for cancer getting worse. Good?
Isn't one estrogen one of those hormones that gets like released into water supplies and food?
I don't believe so.
Dave?
No?
Yeah.
Because the steroids put into the food supply?
Right.
All right. Okay. I don't know much about that, but okay.
Okay. No, it's just an interesting aside.
All right. So as you can imagine, because this pathway is so important to estrogen receptor positive breast cancer, there have been drugs designed to target it. Next slide. One of the major classes of drugs that targets the estrogen receptor pathway is selective estrogen receptor modulators, or SERMs for short, such as tamoxifen, which is the drug I mentioned earlier. This is what tamoxifen looks like. Basically what happens with tamoxifen is it's broken down in the liver into 4-hydroxyl-tamoxifen, which then it acts as a competitive inhibitor to estrogen with respect to the estrogen receptor. Now what that means is that basically this 4-hydroxytamoxifen mimics estrogen chemically. It's very similar to it. And it attaches to the estrogen receptor on these cells. And because there are only a limited number of estrogen receptors, that means that estrogen can't attach to those estrogen receptors and stimulate the bad stuff associated with cancer. Make sense?
One more time.
All right. So tamoxifen gets broken down in the liver and into 4-hydroxytamoxifen. 4-hydroxytamoxifen attaches to the estrogen receptor. Estrogen can't attach to the estrogen receptor, and you don't get the bad stuff associated with cancer. Better? Yep.
So is tamoxifen the drug that you were using kind of during your
Yeah.
during your research?
Can we get everybody that's standing just to come in and sit down and relax?
Yeah.
You guys are uncomfortable.
Where?
On the floor.
I'm pretty sure.
Yep.
Does tamoxifen does it attach to estrogen receptors in other places?
It does, in fact. Actually, that's one of the other problems with tamoxifen is that tamoxifen acts as an antagonist to this pathway in some tissues, but in other tissues, it actually acts as an agonist to make this pathway worse. So it can actually result in cancer in other parts of the body, such as endometrial cancer and a few other things. Yeah.
But isn't estrogen an important hormone in the body, so wouldn't not allowing it into the cell cause a lot of problems?
This doesn't not allow it into the cell. This works entirely within the cancer cells, though obviously if the drug were where the drug sometimes obviously does affect some other cells, but not to a significant degree. There's actually another class of drugs called aromatase inhibitors, which prevent androgens from being converted to estrogen, which basically just prevents almost all estrogen from being created and processed in the body, which has a lot worse side effects in that regard. All right, so SERMs, was there a question? Yeah?
How does the drug specifically target the cancerous kind of estrogens and not the
I don't know too much about that, but there aren't very many tissues in the body that are significantly estrogen responsive. Breast tissue, endometrial tissues, bone tissue. So the drug would only target those tissues in theory because they're responsible for that. Additionally, it only has antagonistic activities in some of those tissues, so otherwise it would function like estrogen, basically, in some other tissues. Did that answer your question? All right. So serum, like tamoxifen, bind attached to these proteins called co-repressors, which are like the cofactors that I mentioned in the previous slide, except when they bind the estrogen receptor, they bind to it permanently, preventing the estrogen receptor from being used again. Additionally, instead of activating transcription factors that would result in the production of those proteins that cause bad things for people suffering from cancer, instead they actually repress translation of those factors. So you don't get the expression of those factors. Yeah?
And those are administered in the drug with when you put tamoxifen.
Those are
in
found
as well.
naturally.
They're naturally.
Yeah. All right, next slide. So, actually go back, sorry, I forgot one piece of it. So the problem with tamoxifen, is that, as I mentioned before, about 33% of patients who are treated with the drug over the course of 5 to 10 years develop resistance to it. This doesn't necessarily mean tamoxifen is a bad drug, however, it just means that we need to better understand how these cells become resistant, and then how we can potentially reverse this resistance. Next slide. So how do cells go from being tamoxifen sensitive to tamoxifen resistant? And what are the molecular mechanisms behind that? Well, the short answer is it's complicated. The precise mechanism is not exactly understood. However, studies have shown that approximately 90% of tamoxifen-resistant tumors still express the estrogen receptor. This is really important because if tamoxifen-resistant cells just didn't have the estrogen receptor, there would be nothing we could do. The drug would just not be functional. But because most tamoxifen-resistant cells do have the estrogen receptor, in theory, we can just, if we can figure out the molecular mechanism responsible for this resistance, then we can just, next slide, then in theory we can convert tamoxifen-resistant cells back into tamoxifen-sensitive cells and resensitize them to the drug. And some research has indicated that this missing link here, this question mark, may in fact be a protein called EIF4E. Next slide. Well, actually first, does anyone have any questions about the material I've presented so far? Yep.
Just a quick question. How long does it take for the body to build up resistance to tamoxifen?
It can happen at any point in the 5 to 10 year period. Mean time is around 3 years, but that's just off the top of my head. Don't quote me on that. Yep.
What's the actual mechanism in the cell that causes the resistance?
I'm gonna get to that. All right, next slide, because we're running out of time. So EIF4E is or eukaryotic initiation factor 4E, is a protein responsible for initiating selective translation of certain mRNAs in the cell. So that's complicated. What is selective translation? Many of you probably know that translation is the process by which a ribosome reads a piece of messenger RNA and then creates a corresponding protein copy. Proteins are responsible for performing many of the functions of the cell. Without translation, cells couldn't live because they couldn't make proteins. So there are two main types of translation in eukaryotes in cells like humans. Global translation and selective translation. Global translation is the equal translation of all messenger RNA that comes to the ribosome. I say equal because it doesn't preferentially translate any one particular mRNA over another. And this is very good for when a cell is trying to maintain its existence or trying to maintain a current state. However, when there's some kind of emergency, such as say you get a cut, the body will rely on selective translation. The selective translation allows the cell to create large quantities of a particular protein very rapidly. So for example, in the case of a cut, you might see skin cells create factors that help resist cell death or encourage cell proliferation, both of which are important parts of the wound healing process. However, those same phenotypes, those same things, are also commonly seen in cancer, which is by definition a disease of uncontrollable cell proliferation. So selective translation is commonly misregulated or dysregulated in these cancers. And since EIF4E is one of the main proteins responsible for initiating or causing selective translation, it seemed natural to look at it in tamoxifen resistance. And because EIF4E has the capability to create proteins that could be potentially very damaging to the cell and the body, it's very highly regulated. Any questions about EIF4E before I go on to explain how it's controlled? Okay, excellent. So this very complicated diagram tells you exactly how it's controlled. I want you to ignore everything that isn't in that red box because it's very complicated. But basically there are two main pathways or two main sets of proteins that are responsible for controlling EIF4E. The PI3K AKT mTORC1 pathway, which you see here on your left, is the first of them. And basically, the first protein in this pathway is 4EBP1, which is right there, which binds to EIF4E and sequesters it, basically prevents it from initiating selective translation and creating all of these things which are bad for cancer, which are good for cancer cells and bad for the body. 4EBPs are inhibited by a protein complex called mTORC1. So mTORC1 prevents 4EBPs from attaching to EIF4E and thus letting all that EIF4E go out and do selective translation and cause bad things to happen in the cell, encourage cell proliferation, growth, and survival. So naturally, because this pathway presents such an interesting target, a drug has been developed that targets this pathway by the name of everolimus, or RAD-001. Everolimus recently reached phase 2 clinical trials, and everolimus works by inhibiting mTORC1. So, to get this straight, everolimus inhibits mTORC1, which inhibits 4EBP1, which inhibits EIF4E. So everolimus basically, eventually down the line, inhibits EIF4E, is what you should take out of this.
Which resensitizes the cell.
Yes, which in theory should resensitize the cells to tamoxifen. Yep.
The fact that EIF4E even exists in the cell, it has to have some sort of other functionality in healthy bodies, right?
As I mentioned, selective translation is necessary for survival. Like as I mentioned, if you have a cut, you need to perform selective translation. Selective translation is often used as a response to changes in the environment.
Do you know what that protein is specifically?
We don't know all the targets of EIF4E, but a lot of them are proteins associated with cell survival and proliferation. Good? Did that answer your question?
Yeah.
Okay. So basically, RAD-001 prevents EIF4E from functioning. And the problem with RAD-001 is that though it's effective, it has some serious side effects such as stomatitis, which is swelling of the mouth, and immunodeficiency, because the mTOR complex over here is also involved. Yeah.
No.
Oh, okay. Okay. Is also involved in regulating the immune response. So it's only given generally as a treatment, kind of as a last resort. However, as some of you may have noticed, there's another side to this pathway. And that is the RAS-MAP-RAF-MEK-ERK pathway, which is kind of hard to pronounce. Basically, the other side of this pathway over here. So there's this protein called MINC1, or MAP kinase interacting kinase 1. And MINC1 phosphorylates EIF4E. It attaches the phosphate group to EIF4E. And phosphorylated EIF4E is hyperactive, meaning that it's capable of capable of translating more of these EIF4E sensitive mRNAs that result in cancer becoming worse. So phosphorylated EIF4E is worse for cancer. So what we did is we wanted to look at the levels of phosphorylated EIF4E in tamoxifen-resistant and tamoxifen-sensitive cells certain amounts of time after treatment with tamoxifen. So we performed a Western blot, as you can see up here, and then a bar graph quantifying it. Western blot just measures the amount of certain proteins in the cell. So we looked at the levels of total EIF4E here on the bottom, and we found that they were about the same in tamoxifen-sensitive and tamoxifen-resistant cells. There wasn't a significant difference between the two. However, the proportion between phosphorylated and the percent of the total EIF4E that phosphorylated EIF4E represented, basically the amount of hyperactive EIF4E was significantly greater in tamoxifen-resistant cells as opposed to tamoxifen-sensitive cells. So basically, EIF4E hyperphosphorylation or hyperactivity is correlated with tamoxifen-resistant breast cancer cells. Does that make sense? Yep. Yes, basically. There's more of the bad version than it's. I'm gonna have to keep moving because the next slide. We're running out of time. So then what we did is we decided to treat these tamoxifen-sensitive and tamoxifen-resistant cells with a drug called CGP. CGP is a drug that is designed to inhibit MINC1 and prevent MINC1 from working. So what we did is we took tamoxifen-sensitive and tamoxifen-resistant cells and exposed them to a variety of concentrations of CGP and measured the levels of phosphorylated EIF4E, regular EIF4E, and beta-actin, which is control in this case. And we found that in both tamoxifen-sensitive and tamoxifen-resistant cells, CGP had no effect on the levels of total EIF4E because CGP doesn't interact with EIF4E at all. But we found that in both tamoxifen-sensitive and tamoxifen-resistant cells, treatment with CGP decreased the amount of phosphorylated or hyperactive EIF4E significantly in both cases, although a higher dose than placebo required tamoxifen-resistant cells. Basically what this implies is that MINC1 is phosphorylating or making hyperactive EIF4E in human breast cancer cells that are resistant to tamoxifen. And CGP is effectively inhibiting MINC1 and preventing that. Does that make sense? Next slide. So what we then did is we wanted to see if treatment with CGP was a viable way of treating this tamoxifen-resistant breast cancer. So what we did is we looked at colony formation. For those of you who don't know, colony formation is a good indication. It's the ability of cells to form colonies when you put them on a petri dish and provide them. So if you put normal human cells on such a dish, then they won't do anything. They won't grow, they won't form colonies because their proliferation, their division is very regulated. Cancer cells, on the other hand, will grow rapidly and form these colonies. So basically, colony formation is an approximation of the ability of cells to form tumors in vivo. So more colony formation means that you have a worse cancer generally. So we treated the cells with either DMSO, 1 micromolar tamoxifen, which is a standard dose given in treatment, 10 micromolar CGP, which is the dose we determined was effective in the previous slide, and then the two in combination. So as you can see, tamoxifen-sensitive cells responded well to significantly decreased colony formation in both CGP treatment and tamoxifen treatment. But there was an even more significant decrease in colony formation, to about 10% actually, in cells treated with both tamoxifen and CGP, which is suggesting that the two drugs work very well in combination. When you look at the resistant cells, you see obviously they don't respond well to tamoxifen. That's what makes them resistant. But these tamoxifen-resistant cells do respond to CGP treatment and respond much better to CGP and tamoxifen together than they do just tamoxifen alone. Does that make sense to everyone? Does anyone have any questions? How many people are lost? All right. Last slide. Just go through all the bullets. That's just basically a summary of what I did, of what I presented on so far. I would read it to you, but we're running out of time, so I'll just go to the last bullet, which is what my mentor is doing next. So what he's planning is to treat cells with this so-called triple treatment, a treatment of CGP, RAD001, and tamoxifen. This is, in theory, going to be really effective because both CGP and RAD001 will help to knock out, to prevent EIF4E from properly functioning. And then tamoxifen will be able to kill the cells like it normally does. And this treatment shows great promise, and it's going to be done in animal tumor modeling. So hopefully it will be an effective drug treatment and help give hope back to those 33%. Thank you.